Introducing the Central Dogma with your amazing, intelligent, spectacular, amazing host, the dog named Ma. Hello. As you already know, I'm the dog named Ma, but you can just call me Dogma. Today I will be teaching you about the super fun, super complicated topic known as the Central Dogma. Now, by definition, the Central Dogma provides a basic framework for how genetic information flows from a DNA sequence to a protein product inside the cell. That sounds kind of complicated, because it is. However, the Central Dogma can simply be described as the flow of genetic information. This flow begins with DNA replicating itself, turning DNA into RNA through transcription, and turning RNA into proteins through translation. All cells express their genetic information this way and must control and regulate the efficiency and the amount of proteins necessary according to their demand. Let's begin with replication. DNA replication is the process in which the original DNA template strand replicates or produces two identical daughter strands. Replication begins with the enzyme helicase. Helicase unwinds the double helix nature of the original DNA template strands, creating a replication fork in the DNA. This unwinding action is kept in place by the single-stranded DNA binding proteins. These proteins keep the DNA from going back together. We are working with two strands, a leading strand and a lagging strand. Let's begin with the leading strand, which codes in the 5' to 3' direction. Replication occurs extremely easy on this strand. DNA polymerase III moves along the replication fork and adds nucleotides in a continuous fashion. The lagging strand, on the other hand, is discontinuous, since it is being coded in the 3' to 5' direction. RNA primase lays down RNA primer. DNA polymerase III lays down short segments of DNA, creating Okazaki fragments. DNA polymerase I replaces RNA primer with DNA. Then the DNA ligase links all the separated fragments together. DNA replication is indeed fascinating. However, it is not always 100% perfect. There are various mutations that can occur throughout DNA replication. The first type of mutation we will discuss is substitution or point mutation. These are mutations that alter the one nucleotide in the DNA sequence. Let's look at the triple codon AAC, for example. AAC should code into TTG, which then codes into RNA AAC. The amino acid for this would be asparagine. However, if AAC accidentally coded into something different, like CTG, then the RNA sequence would be GAC, and therefore the amino acid would be aspartic. Mutations that result in a different amino acid are called missense. Let's take a look at another example, TAG. TAG should code for ATC, which would then code into the RNA sequence UAG. If TAG accidentally coded into ATT instead of ATC, then the RNA sequence would become UAA, which would result in a stop codon. We call these premature stop codons nonsense. Lastly, let's look at the DNA sequence TAT. This would code into ATA, which would then code into UAU. It just so happens that TAT accidentally codes into ATG, which would code into the RNA sequence UAC. Luckily, both UAU and UAC produce the same amino acid, tyrosine, so no damage is really done. We call these silent mutations because nothing is really bothered. There are two more mutation types, deletion and insertion. Let's start with deletion. If we have the RNA sequence GGT, CTC, which codes for the tRNA sequence CCA, GAG, the resulting amino acids would be proline and glutamic. However, let's say that the first C was accidentally deleted in the RNA sequence during transcription. If this happened, then the next nucleotide in line would be part of the triple codon. In this nucleotide is cytosine, or C, it would code into G, and the entire amino acid would be glycine. Insertion is a similar process, except a nucleotide is added instead of removed. If adenine is added after the first C, then the triplet codon will change from CTC to CAT. The end result is an amino acid of aspartic. These two mutations are known as frame shift mutations since the adding or removing of the nucleotides shifts the total framework of the sequence. Overall, replication is a never-ending process of DNA dividing itself into two more strands. The next process is transcription, or the process of newly replicated DNA being turned into RNA. Let's begin with the initiation complex of transcription. Here we have a strand of DNA. The promoter is a region of DNA that signals the gene transcription. Promoters are located near the transcription start sites of DNA strands and can be 100 to 1,000 base pairings long. The start point is where the DNA will start its coding for RNA. This is done by the enzyme RNA polymerase. The template strand of DNA is the strand that gets copied. It is called the template strand because it acts as a template for the mRNA assembled by ribosomes. Sequences on the template strand are complementary to the mRNA sequence. The non-template strand already contains the gene since it is moving in the 5' to 3' direction. The Tata box is a DNA sequence in the promoter region that only contains A and T nucleotide bases. This sequence helps to determine where the transcription will begin. The Tata binding proteins bind to the Tata box, and several transcription factors bind to the DNA itself. These bindings form an initiation complex of the transcription. RNA polymerase essentially opens up a segment of the DNA to assemble a new chain of nucleotides to produce a complementary RNA strand. This can be summarized as elongation. RNA synthesis continues to function along the template strand until RNA polymerase reaches a stop signal. This signal tells the strand to form into a hairpin loop so no more coding can be done. This is the final process called termination. Let's take a more in-depth look at the RNA coding process. Here we have our DNA strand. Let the blue dots represent adenine, the green dots tyrosine, the orange guanine, and the yellow cytosine. Each nucleotide matches up with its complementary pair. However, RNA polymerase comes in and transcribes this DNA into complementary RNA strand. Instead of adenine pairing with tyrosine, it is now paired with the RNA base uracil, represented by the pink dots. This coding continues until RNA polymerase reaches a signal to stop, and then the RNA strand is formed. Our last topic is translation, the process in which RNA codes into amino acids to form a polypeptide chain. As we already know, the original DNA strand codes into mRNA, which then codes into tRNA. However, this process is still part of transcription. Translation is when the tRNA translates into an amino acid based on each set of 3 nucleotides. These codes can be found using a codon chart. Transcription is a fairly simple process. tRNA is bound to an amino acid and links to the appropriate mRNA triplet, dropping the amino acid as it goes and leaving empty-handed. This entire process happens in the cytoplasm with the ribosomes that continue to move in one direction. This results in the final product of a protein. Science, biology, the Central Dogma.